Michael has said in his introduction, we are using gene therapy in two different perspectives. We are using gene therapy for the purpose of disease modeling, so trying to generate animal models that are better than the ones we are currently available. And on the other hand, of course, we are also using gene therapy for therapeutic purposes. So what I'm going to talk most of my part today is the use of viral vectors for generating better animal models. So one thing that most of us we know and we have been suffering for a while is that we have to recognize that most of the animal models we have at our disposal are far from the ideal. So most of these animal models only reproduce a small part of the disorder. Most of these animal models don't have the adequate phenotype. So overall, they have plenty of limitations. So not surprisingly, when the preclinical pipeline is being tested in these animal models, when coming to clinical trials, most of these clinical trials always failed. So there is room for improvement. And I think that we need to really keep an eye on how we can generate better animal models. And in my field, that is Parkinson disease, there are a number of hallmarks of the disorder, particularly when coming to reproduce the non-neuropathology of the disorder. So in humans, the substantia nigra is pigmented. It's pigmented because there is a pigment called neuromelanin. That is what gives the name of the substantia nigra. It's not the green substance, it's just the black substance because of the content of neuromelanin. So only primates, no matter these are human or non-human, they have a natural accumulation of neuromelanin over time. In Parkinsonian patients, what is the diagnostic criteria in terms of neuropathology of the disorder is that the neuropathologists need to find Lewy bodies in the substantia nigra, and these Lewy bodies are observed in pigmented neurons. So here just a couple of examples. This is an stain for alpha-synuclein immunohistochemistry at the level of the substantia nigra in a patient, in a human patient. This is neuromelanin, and you can see this neuron has a couple of Lewy bodies that are positive for alpha-synuclein with this typical appearance of a peripheral halo that is more intense reactive against synuclein and a paler center. There are a number of other markers, such as P62 is a marker for autophagy, but very good for disclosing the Lewy bodies. So this is another patient suffering from idiopathic Parkinson disease with two pigmented neurons, one with up to 5 Lewy bodies, the other one with not a single Lewy body. This is another case. It was an individual suffering from a genetic form of Parkinson disease caused by a glucocerebrosidase mutation, where you have two pigmented neurons. This is neuromelanin, and both neurons have a number of Lewy bodies. So the point is that when coming to design animal models, this is the type of neuropathology that we need to reproduce in order to have an animal model that is relevant enough. So this project was built on the former expertise of Miquel Vila. For those of you that don't know, Miquel has implemented a very smart strategy by taking advantage of an AAV coding for the human tyrosinase gene, that is the enzymatic precursor of neuromelanin. So upon delivering the substantia nigra of rats, and this substantia nigra becomes pigmented, Lewy bodies start to degenerate, and so on. So at the beginning, what we just did is just to upgrade Miquel's model in rats up to non-human primates. So as you can see, we deliver the AAV coding for human tyrosinase into the nigra. These neurons get pigmented over time. At some point, they exhibit Lewy bodies, and once these neurons die, neuromelanin goes extracellular, and so on. So this is what we have done in the past. You can see pigmentation of the substantia nigra in these monkeys, even macroscopically. So this is pictures taken with a cellular phone on the microtome, where you can see that the entire substantia nigra from rostral to caudal levels became pigmented. In the left substantia nigra, this is where we have delivered the AAV, control AAV and the contralateral substantia nigra. And the entire substantia nigra is pigmented. This is from an animal, sorry, my secretary calling. So this is a substantia nigra for an animal with a follow-up of 4 months. I mean, this is the results in the outcome of 4 months of follow-up, 4 months after the delivery of the AV. And if you wait a little bit longer, you can see that the substantia nigra is still pigmented, but there is a loss of pigmentation over time because these neurons are about to die over time. It's exactly the same thing, the same amount of pigmentation that you may expect in human beings. So, well, this is just to show you neuromelanin in 4 different animals, the number of neurons that became transduced, and blah, blah, blah. It's not that much important. This is the type of lesion we can induce. We have a much better lesion in 2 females and not that much in 2 males. And this is the type of inclusions we can see. So the message here is that you don't need to spend the entire day behind the confocal looking for a Lewy body. There are plenty of intracellular inclusions that are p62 positive, also positive for alpha-synuclein. Inclusions are made of 2 types. When located in the cytoplasm, these are Lewy bodies, and when located in the nucleus, these are Marinesco bodies. Marinesco bodies are an indirect indication of accelerated aging. So this type of Lewy bodies that are observed in pigmented neurons are positive for all type of species of alpha-synuclein. So this is antibody against total alpha-synuclein. This is digested with proteinase K, so we get rid of soluble alpha-synuclein, and this is just insoluble alpha-synuclein that is also phosphorylated, as can be seen with a different antibody. Of particular importance, 2 things. First, inflammation. So once these neurons die, neuromelanin goes extracellular and is often observed in perivascular locations. As you can see here, this inflammatory reaction is made of activated microglial cells that are also CD68, so it might be a role for macrophages here. But you can see these neurons contain intracellular neuromelanin. What else? And since we are dealing with endogenous alpha-synuclein, so we are not about to induce, we are not enhancing synuclein with an AAV, we are just enhancing neuromelanin accumulation. Synuclein is something coming later, and it's made of 100% endogenous forms of alpha-synuclein. So we took a look at the cerebral cortex in an attempt to evaluate to what extent we can see a spread, anterograde spread of alpha-synuclein. And the quick answer is that yes, this is a pyramidal neuron receiving a dopaminergic fiber and going to this neuron. And you can see this an inclusion that is p62 positive, just opposite in the postsynaptic element. This is another example. I think it was superior angular gyros in that case. This is the TH fiber coming, p62 positive, and alpha-synuclein positive. So I'm going a little bit quickly because I don't want to spend that much time in this part of my talk. Just to summarize, the main important message is that there is evidence. We don't know which is the ultimate mechanism, but we managed to provide evidence that the synucleinopathy is being triggered by the accumulation of neuromelanin. We have observed Lewy bodies for the first time in non-human primates, and we can also demonstrate that this is an anterograde expression of alpha-synuclein. And this is my PhD student, Julia, very happy for her first publication that has been recently accepted in brain. Okay, so what else? And this is something we don't have discussed before. So at the beginning, we have a feeling that we were able to see more and more Lewy bodies in the ventral part of the substantia nigra compared to the dorsal part of the substantia nigra. At the beginning, it was just nothing but a feeling. However, Julia spent a considerable amount of time in trying to locate the right position for every single neuron that is pigmented with Lewy bodies. So these are 4 animals from rostral to caudal section of the substantia nigra. What you have in these gray dots are pigmented neurons. What you have in green are pigmented neurons with Lewy bodies. And what you have that are positive for aldehyde dehydrogenase is a marker for the ventral tier of the substantia nigra. And what you have in blue or in purple are neurons in the dorsal tier of the substantia nigra positive for calbinin and Lewy bodies. So, the distribution is quite evident, but indeed, if you count the number of Lewy bodies from animal to animal, in the end, there are more than 90% of Lewy bodies are located in neurons of the ventral tier that we know that by far are the most vulnerable ones. So you go behind the microscope with more details. So this is just a coronal section of the substantia nigra. This is the midline. This is the substantia nigra. This is the red nucleus, for example. This is stained with aldehyde dehydrogenase type 1. This is a marker for the ventral tier of the substantia nigra. And this is the next section. In that case, it stands for tal binding. You can see also the red nucleus here. This is the midline. This is the substantia nigra. Tal binding is a very good marker for the dorsal tier of the substantia nigra. So, even at low magnification, I hope you can see that there is a stripe of pigmented neurons that are located in calbinding pore area. So it seems that it's a preference for neuromelanin to be located in areas that don't have calbinding. And if you go to higher magnification, no matter you are dealing with the substantia nigra pars compacta or with the VTA, you can see that neurons positive for the marker of the ventral tier, they have much more neuromelanin than neurons in the dorsal tier. And it's not only more neurons being pigmented, but also neurons being pigmented have a much higher levels of neuromelanin. And with this level of magnification, even in bright field, you can even see intracellular inclusions. Of course, they are not positive for anything, but this looks like really low bodies in these neurons. However, you just take a look at neurons that are calbinding positive. There are just a few of these neurons that became pigmented, no matter you are dealing with the substantia nigra pars compacta or with the VTA. So you compare calbinding positive neurons being pigmented with neurons in the ventral tier being pigmented. The difference is quite striking. Of course, it needs to be quantified, but the difference is pretty clear. So if you go behind the confocal microscopy, just trying to disclose the different type of neuronal populations, a typical example is a neuron in the ventral tier that is positive for this marker, is negative for calbinding, has a liquid body, and is heavily pigmented. This is what we found more often. Only very few neurons that are calbinding positive and negative for aldehyde dehydrogenase, they exhibit liquid bodies. Only very few. And here you have the ideal example. You have one neuron that is heavily pigmented, is positive for aldehyde dehydrogenase, so it's a neuron of the ventral tier. There's another neuron here that is positive for both aldehyde dehydrogenase and calbinding, but with a very small amount of neuromelanin here. And for sure, these neurons don't have any liquid bodies. So we have two neurons close to each other. The one that is heavily pigmented is positive only for this marker, and it's the only one that has a liquid body. What else? So at this point, a question that I'm going to come back later on, but it's something I want that you keep inside your hippocampus, because we are going to be back again to this question later on. So to what extent we can correlate three different things: the content of calbinding, levels of pigmentation, and the presence of liquid body pathology. So I think these three items, and they are doing together like a synergistic effect, but we will be coming back later on to this question. Just keep in mind. So what we did next, as a different strategy, instead of delivering AAVs in the substantia nigra, we are changing the AAV serotype, and we are now using an AAV9 instead of an AAV1. The main advantage of AAV9 is that, at least in non-human primates, is transported in the retrograde direction very efficiently. So by taking advantage of brain circuits, the message is that you can induce transduction over some distance if you are dealing with areas that are interconnected. So if you are, delivering into a given brain area an AAV9, all brain areas that are innervating the area of deposit are going to be transduced by retrograde spread of AAV9. So we have delivered AAV9 coding for human tyrosinase into the putamen of these animals. Some of these animals with unilateral injections, few other animals with bilateral injections, and this is just the readout of the macroscopy readout. So this is just one single animal for very rostral to very caudal levels of the nigra. Of course, this animal has been injected unilaterally in the putamen. You can see there's only pigmentation in the left substantia nigra. This other animal, by contrast, has been injected in both the left and right putamen. So that's why pigmentation is bilateral. and it's also, being expressed neuromelanin in the entire rostrocaudal extent of the substantia nigra. So it's another way of having access to the substantia nigra, with the added value that every single neuron that became pigmented, and we know which is the identity of this neuron. So this neuron is going to innervate the putamen, because this is the only way by which this neuron can become pigmented. Well, this is just some histological readout. What you have in the first two rows on the top is an animal with a unilateral injection, so there's nothing in the right substantia nigra. There is some evident pigmentation in the left substantia nigra. And these are the animals with a bilateral injection of this vector in the putamen. You can see there is a bilateral pigmentation both in the left and in the right substantia nigra. With this type of material, once you go behind the microscope, you can disclose pigmented neurons, more or less neuromelanin, that are Th positive. These neurons have intracellular inclusions that are positive for p62 and for alpha-synuclein. So there are very different, a wide variety of examples of, for instance, here you have a neuron with even 3 inclusions that are all positive for p62 and alpha-synuclein. We have managed to see neurons with up to 7 Lewy bodies. And this picture was taken here because we are trying to address a different question. There might be some people that argues that the core of the Lewy body is made of a neuromelanin grain. So just by chance, here we have a neuron with a Lewy body that is positive for synuclein, p62 is Th positive. But here you can see in bright field that the texture of the Lewy body is completely different of a neighboring neuromelanin grain. So at least in our non-human primates, we never have a chance to see a Lewy body with a neuromelanin grain inside. So, and this is another thing. We are having a lot of fun, by dealing with this new AV. So, in brief, and this is something that I think there are not that many of you dealing with non-human primates, but for sure there are most of the people are dealing with mice. This has been designed to be used in mice, and this is what I want to emphasize in my talk to let you know. This is available, and just in case you want to try, just give me a call. So this is a new AV. There is, there are continuous race for the development of new AV capsids. But this has been published quite recently. It's coming from a company based in Cambridge called Voyager Therapeutics. Voyager Therapeutics have a lot of interest in Parkinson's disease. This is a company that was pushing forward gene therapy with topical carboxylase until phase 2. So they have quite a nice expertise in the field. And, by going to a super sophisticated procedure called tracer, don't ask me to talk about it because, I don't understand more than half of the paper. In brief, they managed to generate a library of new capsids that can penetrate the blood-brain barrier upon systemic delivery. And they have generated up to 10 or 12 capsids. From those in the paper, the type of AV9 capsid variant that looks the most promising one was called P31. So you want to go through the paper, don't pay that much attention to the enormous technology behind. Just look at the images, and I can tell you AV9 P31 variant is doing a very nice job. So before getting started, we have synthesized this capsid using a standard reporter such as GFP, just to test to what extent we can reproduce the findings in the paper, because sometimes you get too enthusiastic about, and once you try it, it never was the same. So we just want to give a try to see to what extent we can reproduce the original findings of the paper with GFP as a report. And the answer is yes, it was really, really very nice. So what you have here on the left is a mice brain injected systemically with this P31 AV9 variant coding for GFP. So this is GFP immunohistochemistry. You can see that the entire brain is transduced. It has an ubiquitous promoter. So you have the entire cortex transduced, neurons and glial cells, nice labeling the hippocampus, brainstem, nucleus basalis of Meiner, and the cerebellum. So there are, you can even see Purkinje cells that are entirely transduced with the whole dendritic tree. And this is, for the same animal, this is the liver. Because you can see a number of hepatocytes here that are positive for GFP. But, when dealing with people that are specialized in gene therapy in the liver, they will say that this is a minimal transduction in the liver, at least compared to a standard naive AAV9. So with an AAV9, this will be, almost 100% of the hepatocytes being transduced upon systemic delivery. So this is not that much compared to what can be expected for an AAV9. So this is an AAV that is directed against a receptor that is only present in the BBB of mice. We try in rats and it doesn't work. We try in non-human primates, it doesn't work neither. So it's only for work in mice, but it works very nicely. So once we managed to see that the penetration of the BBB was optimal, we came back to our original strategy and we have decided to mount on the P31 cassette. Instead of GFP, we have decided to mount in the human tyrosinase gene. And this is the readout. So we have 2 cohorts of mice made of 4 mice each. So for the first cohort, we have delivered IV this AV9 P31 human tyrosinase. And we sacrificed these 4 mice with a follow-up of 1 month. And later on, we sacrificed the second cohort with a follow-up period of 4 months. And this is the substantia nigra. So a low magnification, there is not that much to see. This is staying with Mason Fontana to enhance neuromelanin visualization. However, if you go a little bit more into details, you can see that, as short as 1 month post-injection, there is some pigmentation going on in the substantia nigra. You need to be aware that rodents don't have a trace of neuromelanin. So if you have a chance to see neuromelanin, this is just because of the transduction with the AV. So you can see a number of pigmented neurons. They are not that much heavily pigmented in the substantia nigra and the VTA, but don't forget that this is just with a follow-up of 1 month. If you wait for 4 months, the levels of pigmentations are very, very high, both in the substantia nigra as well as in the VTA. So it looks appealing. At least it's up to you to think about which transgene you will mount on this cassette. So you can mount tau, you can mount alpha-synuclein, you can mount whatever you may have in mind. But at least, as far as tyrosinase is concerned, it's doing a pretty nice job. So, of course, you are about to express tyrosinase all over the brain. But the main advantage is that only catecholaminergic neurons are going to be pigmented, because you need to be of a catecholaminergic phenotype to be able to accumulate neuromelanin. This is some results at the level of the locus coeruleus. That is another catecholaminergic center. There's not that much to see at the follow-up period of 1 month. I don't know, you can see, for instance, it's just one single neuron, maybe 2, with a tiny little amount of neuromelanin. I know it's hard to see. However, if you wait a little bit longer, I think that now you can appreciate that there are a number of neurons here in the locus coeruleus that start to be pigmented. Not at the same level of pigmentation as in the substantia nigra. So it seems that the substantia nigra and the VTA with the same survival period are going to be much more heavily pigmented than the locus coeruleus. But, at 4 months, most of the neurons in the locus coeruleus will exhibit a mild amount of neuromelanin. And there's another place that is worth to take a look, is the dorsal motor nucleus of the vagus, that in humans is also pigmented, not pigmented enough to be visible to the naked eye. But if you go behind the microscope in a necropsy material, you can see some pigmentation in the dorsal motor nucleus of the vagus. Don't forget that the dorsal motor nucleus of the vagus, locus coeruleus, and the nigra are the 3 brain nuclei that have been engaged in the propagation of Parkinson's disease according to the Bragg hypothesis. So for the animals with a 4 months period of follow-up, I hope you can see here that there are not that many neurons pigmented, but there are quite a few. Pigmentation is not too heavy. We are not expecting to see, something supreme. But, the message is that, don't forget that this is an AV being injected systemically. So it's quite impressive to see there are some neurons in the dorsal motor nucleus of the vagus that also exhibit a small amount of pigmentation. Okay, so you go behind the confocal. For these neurons that became pigmented, at least with a follow-up of 4 months, you can see Lewy bodies that are positive for p62, alpha-synuclein, in Th-positive neurons that became pigmented. So we have pigmentation, we have Lewy body pathology. We don't have neuronal death so far with a follow-up of 4 months. We will see, but so far at 4 months there is not any relevant or detectable level of Th cell loss. Here what is shown here. So this is Th in the substantia nigra. This is Th in the caudate putamen. This is lateral substantia nigra. Substantia nigra pars compacta, VTA on the left side, VTA and substantia nigra pars compacta on the right side. I hope that you will be able to see that most of these Th positive neurons are pigmented, both in the substantia nigra pars compacta and few of them in the VTA, and the same on the other side. So coming back to my former question, just keep an eye on this. So what you have on the left is one section of the substantia nigra of these mice stained for aldehyde dehydrogenase to disclose neurons in the ventral tier. And this is the next section, in that case stained for cal binding to disclose the dorsal tier of the substantia nigra. And this is just, some intermediate level of magnification. But if you go to higher magnification, the message here is quite clear. These neurons that are positive for aldehyde dehydrogenase, so these neurons that are located in the ventral tier, are the ones that are more heavily pigmented. And on the other side, neurons that are cal binding positive have a minimal level of pigmentation, if any. You can see that most of these pigmented cells are located in cal binding free areas. So we just give a try on human material. For instance, this is a human mesencephalon. This is the right nucleus. This is the midline. This is the substantia nigra here. You can see that a substantial number of pigmented neurons are located in cal binding free areas. And, quite the opposite is observed if you stain with aldehyde dehydrogenase. So for some reason or another, there is a relationship between levels of pigmentation and the absence of cal binding. And this is just a cartoon that is not that much elaborated, but is good enough to illustrate which is our current thinking. So you just sorry, just take a look at the substantia nigra. You have two types of neurons, those located in the dorsal tier, those located in the ventral tier. So neurons in the dorsal tier are cal binding positive. They became pigmented, but not that much. It has a very mild level of pigmentation. And because of this small level of pigmentation, this low level of pigmentation, the neurons remain alive and are much more resistant to degenerate. You go to neurons in the ventral tier, these neurons are much more heavily pigmented. And because these neurons are above a given threshold of pigmentation, these are the only neurons with a chance to develop Lewy bodies. And because of this Lewy body pathology, in the end, these are going to be the neurons that are about to die. So, just to summarize, the main message is that at present, we think that there is a synergistic effect, ultimately driving neuronal vulnerability in the substantia nigra, that is made of 3 partners. So on one hand, these neurons can be cal binding positive or cal binding negative. Those that are cal binding negative, we know, are much more vulnerable. On the other side, as a second partner, levels of pigmentation. So the more neuromelanin you have in your cytoplasm, you are much more prone to develop Lewy bodies. And that they are, the third partner in this equation. So if you don't have cal binding, you have more neuromelanin, you have more Lewy bodies, you are more vulnerable. And on the other side, if you have cal binding, your levels of pigmentation are going to be much smaller. And because of this small level of pigmentation, you will never develop a Lewy body, so you will remain alive. So for the part of the team that is in the, let's say, in the therapeutic branch, we have 2 ongoing projects. The first project is dealing with Parkinson. Parkinson is an interesting mutation, and there are only very few amount of people with a Parkinson mutation, but this is the mutation that is more frequently observed in patients with an early diagnosis before 50 years of age. It's an autosomic recessive mutation where Parkinson that is engaged in mitophagy. There is a loss of function of the protein. So that's why, because it's a loss of function, can easily be restored with a normal copy of the gene with an AAV. And we have just injected one animal. We're keeping an eye, has been sacrificed last week. So it's too early to say anything. And we have another potential target that so far I prefer to leave undisclosed, that are more or less in the same rational. And that's all so far. Just to stress and to acknowledge, funding received from CyberDebt at the beginning of the project, because it's not that much amount of money, but at least it's something that allows you to get started and to build a collaborative environment with plenty of friends. So that's the end of my talk. I think we are more or less on time. And now, more than happy to take any questions you may have.
All right. Thank you very much, Lanciego, for a very exciting presentation. So the paper is now open for questions and discussion. So as I said earlier, if anyone is interested in posing questions to Lanciego, please do so by using the raise hand option in your screen or by sending me a message through the chat. If by any chance you don't have a microphone available, send me the question written by chat, and I will read the questions to Lanciego. Okay, so any questions?
Well, there are no questions so far. So Miguel, just give me the chance to emphasize that this BBB penetrant variant of AAVs are available to any of you that might be interested in this. So, of course, and because of our collaboration with Miquel Vila, we are pretty much focused on neuromelanin. But this is just a given type of AAV. So you can use AAV9P31 with any gene you may have in mind. So, for instance, for those of you working in Parkinson, wants to overexpress alpha-synuclein, this is a very good candidate because you can deliver alpha-synuclein just by a systemic delivery of an AAV. Quite the same applies for people that are dealing with tau. You can overexpress tau all over the brain without any trouble, but just simply going systemically. Or, on the other hand, just think about that some of you may be dealing with transgenic mice, for a given gene, and sometimes you need something else to push the phenotype. So you are dealing, for instance, for a transgenic mice with synuclein, you can induce pigmentation in this animal. So you may enhance pathology of synuclein. So there are many different corners and many different angles of research that can be covered in mice with such a nice BBB penetrant variant of AAV9.
All right, thank you, Lanciego. I see we have questions that came through the chat. So the question is from Isabel Lastres, and it says, have I understood correctly, after 4 months of the IV injection of the adenine, there is no death of the TH positive neurons?
Let me see. After no, there is. You mean? I hope that I guess that Isabel was thinking about the non-human primates. So we can see TH cell death in animals with a follow-up of 4 months, as well as in animals with a follow-up of 8 months. I mean, in non-human primates. For some reason we don't know very well, this neuronal death is much more evident in females rather than in males. If you are dealing, Isabel, with mice experiments, after 4 months, we cannot see any evident TH cell loss. Of course, we need to run the appropriate controls, but at least to the naked eye, and this is a quantification that is still pending. But with a follow-up of hormones, we can only see neuromelanin and Lewy bodies, but we cannot see very well there is any amount of cell death in the substantia nigra.
Thank you, Lancy.
Thank you, Isabel.
I just was wondering how old were the mice that you injected?
Well, I think there are mice of 2 or 3 months old, probably no more.
Okay, because in my experience, when you have younger mice, the neuronal death is much
more.
Yeah. So
we are now. It's something I don't have commented that much. I don't have commented at all. But we are now conducting a very dirty experiment. We have delivered AV9 tyrosinase in a pregnant female, and that gave birth to 6 pups. So the overall idea was to check if we can deliver tyrosinase and enhance neuromelanin projections, neuromelanin production in the embryonic stage. So we have injected the pregnant mother, and we have collected the pups, and now we are going to sacrifice a couple of pups every 2 months just to see if we can see any neuromelanin reaching these embryos. In the embryonic stage, we have delivered at E15 just to make sure that these neurons are already postmitotic and that they are not going to further divide. We'll see. There is not that much we have managed to see in the pups 2 months after birth, but it looks like, or it smells like there is a tiny little amount of neuromelanin. So we expect that once these pups gets older, I think we will have quite some very nice chance to see neuromelanin in these pups that has been transduced through the placenta.
Well, for the next seminar.
We'll see. Perhaps we are going to be completely frustrated at the end. We don't know for sure. It's too early to comment. But this is the type of dirty experiment that I think it's always worth to give a try.
Okay, so thank you both, Lancy and Isabel. Any other questions from the audience?
So Ramon Trullas has raised a hand.
Yes, Ramon has raised your hand. Yeah, Ramon, go ahead.
Thank you. Thank you very much, Lance. Extremely interesting presentation. And I just have a question that I may not have understood well. What's your opinion of why the calbindin positive neurons are less susceptible? Is the amount of neuromelanin or something else that is?
It might be something else, Ramon. We don't know. We don't know very well. So in our data, there's some echo. I think so. Okay, so in our data, is this clearly out of the question that calbindin positive neurons are less pigmented. The reason why these neurons are less pigmented is not known. Calbindin, as you know, is a calcium buffer protein, so it's basically just getting rid of calcium. So now we are thinking about why the content of calbindin is making these neurons less pigmented, and we don't know why. But there should be plenty of mechanisms in between, because what is quite clear is that the neurons without calbindin are not only more neurons being pigmented, but also the neurons that are pigmented have a higher amount of neuromelanin compared to the neurons that are calbindin positive. And those that are calbindin negative, we know that for sure they are going to be the most vulnerable ones. But the intimate mechanism linking calbindin content and levels of pigmentation, we don't know anything about.
It's very interesting. Thank you very much. Well, one possibility is what protects the calbindin positive is what makes vulnerable the alkyl binding negative. That would
Yep.
be also very interesting. Thank you very much.
Thank you, Ramon. Miguel, you are mute.
I'm sorry. Yeah, I was saying that we will have time for some questions if there are any. So if anyone is interested, please do raise your hand or send a message through the chat.
No. And otherwise, if there is any question, coming later on, feel free to contact me at any time. Either by phone or by email. I will be more than happy to answer any question that may be coming later on.
All right, thank you. So I don't see any. I still see the Ramon's hands
Right.
still raised, but I guess there is no more questions. It's just yeah. So okay, so this is your last chance for making the questions. If anyone is still interested, otherwise, as Lancy had just pointed out, please feel free to contact him for further discussion or any question regarding the materials or the contents of the webinar and so on. I'm sure he will be happy to
Yeah, for sure, I will.
engage
For sure, I
into
will.
any exchange.
Okay, so thanks
Okay,
a
so
lot
if there
for
are
attending.
Yeah, if there are no more questions, yeah. Thank you, Lancy. Thank you very much for a very nice presentation and for your willingness to share with us very recent data. Thank you everyone for attending, and I hope to see you all at some point in further in future seminars.
Okay.
Okay, have a great
good
day.
afternoon.
Bye-bye.
Bye. Bye. Bye.
Uh,